to Dr. Cho, please introduce you. Yeah, okay. Okay, Carlos, for you. But we will start, it's 9, we are on time. We will start right now, if you agree. Okay, hello everybody, dear friends. Today we have the honor of having the participation of Dr. Cho, Jeffrey Cho, in this webinar to prepare the 7th World Congress on Leishmaniasis in Cartagena, Colombia next year. Thank you very much, Dr. Cho, to accept this invitation. As for the Workplace 7, let me advance a piece of news for you. We have received information that the WHO General Assembly next year will be on the same days that we have for our meeting. And since it is very important that WHO colleagues participate in the Workplace 7, we are making consultation and arrangement to see if we can move forward our meeting until August next year. We will inform you very soon. And Dr. Carlos Costas, a member of the Workplace 7 Organizing Committee, will be the moderator of this webinar and will introduce us to Dr. Cho. Thank you very much, Dr. Costa.
Okay, hello.
Okay.
Hello to everybody. Good morning for some. Good afternoon for many. Good evening for so many on the other side of the world. It's a pleasure. It's an honor to introduce Dr. Jeff Shaw to you. He has been my old friend and professor, of course. He's an extremely bright man since he was a very young researcher in the UK. He's from South London, by the way. And always a sweet man working in the area of taxonomy and ecology of leishmaniasis. He's a deep influence all over the world. Maybe he's one of the best, if not the best, in his field in this area in leishmaniasis. So I'm glad to be here. I'm going to take note of your questions during the presentation. And I'm really happy to introduce Dr. Jeff Shaw to give this speech for you. Jeff, go ahead, please.
Okay. Right, now I have to get my, okay, share, yeah? That it? This one? No, it's the wrong one. Yeah. Well, hello, everybody. Can you all hear me? Okay, thank you. Well, it's good morning to some, good afternoon to others, and good evening to others all around the world. It's a great honor to give this talk, and I've worked on leishmaniasis my whole life, really. And I'm a biologist, and so my focus has been on the parasite. But I never cease to be shocked by the human suffering that this parasite causes. And I've often asked the question, well, why does it do this and where does it come from? I think that one of the things that we must remember is that the parasite is not man. Man is a sentinel animal par excellence. And the disease comes from these little animals, as I pictured here. But some of you who may not be familiar with this may not realize how many animals are involved in the cycle. You can see on this slide, you've got snakes, you've got bats, you've got kangaroos, you have pakas, armadillos. Practically every vertebrate animal has leishmaniasis. But of course, they're not all the same one. They're all very different. But I've always asked myself, what's the fuel that changes, this diversity. And I've come to the conclusion that it's the ecosystems over millions of years that have done this. But I also, another question I asked myself is, well, how did all this begin? Well, as far as we know, the big decision, it was a big gamble. Okay, take another host, because we always believed that the original reservoir was an insect, a phlebotomist or a midge, we don't know. And it took this big gamble, and then it jumped over into a mammal. But it had to do a lot of other things. The big advantage was, of course, that the mammal survives a lot longer than the insect and moves around more. So in terms of benefit, the parasite's gaining on that score. But on the other side, he has to adapt, or it has to adapt to living in a completely different environment. Because in the insect, he's in the intestine, an extracellular one. And then when he got into the mammal, he decided to take up an intracellular one. And we are now finding out that how he does this is absolutely amazing. He manages to live in a cell, he manipulates the cell, he manipulates the host. It's amazing. And also, I've always been interested in the how all this happened. Refinements in DNA molecular clocks have advanced tremendously in the past years. And so have the methods that we look at fossils. Some of you might not know about this, but I'm very interested in this side of it. So now the molecular clocks and the fossil datings are getting more compatible. Now, many of you may not know, but this is the system. We thought that mammals didn't exist with dinosaurs. They do, and they did. And so our dating back has gone right back into the Cretaceous period. And this animal here is quite amazing. The paleontologist did some tests on it and found that it was eating small dinosaurs, which I think is fascinating. The evolution took place over millions of years. And as far as we know, we can go back to about 95 million years in the middle of the Cretaceous period, when the two lines parted. That's the monogenous line here and the digenous line here, which took up the different hosts. And at different periods, it happened that they divided into groups. But the thing is, how did all this happen? Well, one of the theories that now seems to be makes more sense than any other one is the supercontinental hypothesis. This is fascinating because what happened at this period was the different ancestors became distributed as the Gondwandia separated. And so we now have one, which are the most ancient line of leishmania as we know it, the Moundinias. They're all over the world, and we have them here all over the world now, in Thailand, here in Africa, in the Martinique, in the Caribbean, and also in Australia. The other ones are more conservative in their movements. The Biarnias, they stay behind here, and they evolved in Latin America. And the other group, the subgenus Leishmania, their ancestors, we now think that they divide in Eurasia, probably amongst rodents as the rodents expanded. But then something happened. It was terrible. The world almost came to a stop. And everything was, 85% of the animals went, 60% of the plants went. And it took about 15 million years for things to get going again. And this was when the mammals decided to take over, and they became the dominant group. And I think this is where the Leishmania evolved. But we have another group, an ancient group, and I say Leishmaniae because these are all part of a bigger group, and they all are together. And in fact, we now know that parasites of this group, they do actually cause disease. But then the world began to cool, and suddenly the animals became subjected to environments that they'd never had before. And this is, I think, one of the key answers to the distribution of the parasites that we see today, the glacial periods. So they have contrasting situations, epidemiology. In Latin America, predominantly a disease of the forests, although now the visceral form is adapting to the more open areas, but that's man doing that. And in the old world, it's much drier, the whole system. Why could this be that it's drier? Well, could it be that the Ice Age scenarios were different? In the old world, it was very dry, and in the new world, there was still a few refuges around which the parasites could go. So the Ice Age began 2.4 million years ago, and you can't imagine, it was 60 cycle glacial periods. So the environment was changing backwards and forwards and oscillating between cold and warm periods, and the hosts were having to adapt to this, and they did, and so the parasites. Well, this is the last glacial period, and you can see here there's a big difference. Over in the old world, about 18,000 years ago, it's very dry. There really is no forest. But in the new world, in the Americas, we have the four major refuges. And then when it got warmer today, it's still quite dry in the old world. But now we have more forest in the new world. And the secret is that when it got cold, everything went into tropics, more or less, to survive. And so evolution occurred in the tropics, but in the old world, it must have been happening in the desert periods. Now, this is quite fascinating, but when we look at the cold periods, we can see here that the separation of two lines of the New World, the Amazonensis group and Mexicana group, occurred just when the Ice Ages began. So this is really quite a coincidence. Yet the other ones, it all happened. And then man stepped in and he began to distribute the parasites. So we have two factors now, the ecosystem itself and man, who is also doing quite a good job, I think, of distributing the parasites. But one of the things is, well, how does all this happen? Well, I think it happens when we see that that they breed. Well, how do they breed? They breed in different ways, they think, anyway. And the reflection of this is low heterozygosity and high heterozygosity, which reflects inbreeding on one hand and clonal and mixing in populations on the other. Also, there's evidence of hybrids, and we have now hybrids, a lot of hybrids in both the Americas and in the old world. And also, what is dominant? Well, it just depends on the transmission cycle and how the vectors and reservoirs are mixing and in contact with each other. And then we have another method which has been quite popular. It's called mosaic aneuploidy, in which the parasite, he produces a lot of duplicate chromosomes, and it gives him a chance to create new methods of modulation. And this is very useful because in a rapid change, he has a mechanism which he has a better chance of surviving, perhaps than another one. And he hasn't had to do much at all. And this is also employed by plants in the recovery period when they had polyploidy plants. And they think that that was the secret of them surviving after the KT boundary disaster. So genetic analysis on reproduction, the results have to be interpreted with some caution because it depends on the sampling. And this is a beautiful example here of, and I make a plea here because I think that a lot of the work that needs to be done today relies on collections of very good quality. And these are in cryobanks. And the cryobanks need to be supported in the future. This is a multi-locus example in France, and it shows that there's inbreeding. And there was a higher inbreeding in this group, and there was a mixture of transmission here. And they think it's because that the differences are due to differences in the ecology of these regions. So here it's very, very good evidence that these parasites are inbreeding, and this is in the vector. This is in the Andean region, closer to home. And here some lovely work by that's done here by Rougeron and the group. And they showed there that there's also a higher lack of there was the values of the clonality, they were higher, and so there was inbreeding. But if you move these and put them all together, you created disequilibrium, which tended to favor clonality. But it's here. These are all valleys and the whole region, and these valleys are geographical barriers that keep these populations apart. So actually, they don't get the chance to meet each other and create a higher heterozygosity. Now, this is also in an area we've been working in Brazil, in the Northeast. And again, we see quite two contrasting situations here. There's low heterozygosity here, and in this group, higher heterozygosity here. This is a lowland area of forest, and this is an area of Whitmanite, and this area is separated by small valleys, and the whole region was cut into fluvial valleys. But again, this is based on human examples, and so the high heterozygosity may reflect the way of the sampling method, whereas this was in a much more limited population. This is a lovely story. I love this one. You might say, what are these animals here? Well, these are little animals who live in rocks, the rock hyrax. This is feeding on them here, another story. Man did this. He created these rocks, and the hyraxes came, the sandflies came, and this is it. Now, this is all to do with Tropica. I think this is one of my favorite old world parasites because it does everything. It's an anthropogenic leishmaniasis, it does zoonotic leishmaniasis, and in rarely has been associated with visceral leishmaniasis, a very adaptable parasite, Tropica. And this is an example of a beautiful study done based in the Sea of Galilee by Alan and his group. And they showed that actually there was a difference in the susceptibility of the sandfly to different populations. In the northern area, you had Arabicus and Sargenti, and the parasite there, it was adapted to both, and also this parasite from the south adapted to this one. When they took Sargenti though, from here, only the Sargenti parasite from this area only developed here, but the northern one didn't. So this was a very subtle selection of this population to be adapted to this particular population of Sargenti. Again, this is a lovely story, and it came down to the fact that some Petri Vol's group did this, and they showed that it was a difference between the receptors in the sandfly and the ones in the northern population. They had receptors of HV-agglutinin, whereas in the southern ones, it didn't have it. And that is also associated with another story, which is story which you probably have it in having parasites that adapt to this fly that has it and this one that doesn't. But in spite of that, the southern focused Tropica managed to survive in that one. It's a lovely example of how they adapt so amazingly and how so quickly, amazingly quickly. Now, just to have quickly to the Vianias in South America, I think this is perhaps a good example. In the Ice Age, it's 18,000 years ago, we had 4 refuges, basically, the Central American one, the Guiana Shield one, the one in Amazonia, and the one in the southern part of South America. Now, when this Ice Age went, all these forests expanded. And we now find that when we look at these parasites, that the genetic populations of this is SNP class, they've been typed by that they actually, the Amazonian ones, and then you have this group here. And it's quite feasible that this group evolved separately here, that group, the Amazonian ones, evolved separately here, and the two then spread out, so you have mixed populations here. And this happened, of course, long before man. Well, maybe probably long before man actually arrived, which is about 12,000 years ago. I think this is really probably the most amazing piece of research that I really love it. It's it's a piece of research that's gone on between the groups in Belgium, Jean-Claude Jardin, and the people in Peru. And they've studied the different populations of Peruviana and of Brasiliensis. And they modeled it, and then they showed that when in the inter-Ice, last glacial period, that's when everything was much warmer, Brasiliensis was very largely spread here on this side, the Amazon side, and Peruviana, this population here was here, and the other Peruviana here. So two populations of Peruviana, this population and the other population. Then the glacial period came, and obviously the hosts, they went back and they disappeared from this area, and they became very limited to this area. This is where Brasiliensis was. This group then became this particular group of Peruviana became limited, and then this group here remained, and they became less well distributed. Then it warmed up again, and there's the expansion of the Brasiliensis in the Amazon area. This group of Peruviana expanded, and this expanded, and then what happened? You have a lovely hybrid zone here, and they found hybrids there, which is absolutely a beautiful example of how the spreading and the movement of ecological barriers, it creates hybrid zones. The question of hybrid zones is, are these hybrids fertile or not? And that is another story. The last little story, which I think is quite a new one, really, it's an evolutionary force that we don't really understand at the moment. It's a virus symbiont. This is an RNA virus, leishmania virus, very ancient, and it's only known in old groups of protozoa, such as Giardia and Trichomonas, which are very old. It has the same Totaviridae, which is this group. And also it's in fungi. The major Totaviridae, are found in fungi. So there seems to be an extremely ancient relationship between the virus, fungi, and the leishmania, going back, well, millions of years, before the Cretaceous period, maybe. And this is a lovely example of co-evolution. They co-evolve, these different, we have the ones in Bolivia, they have this LRV type, leishmania virus type, and that's a Brasiliensis, that's a different Brasiliensis. Then, and each one has gone with its hosts. And I almost come to the conclusion, perhaps we could identify at least those that have viruses with the virus that they have, but not all of them have the virus. And why they don't have the virus is fascinating. And maybe they actually lost the virus, but how they lost it and why. But they think that the fact that some have a virus could be linked to different pathologies. And the most, for me, the most convincing thing is that immunization of mice with the capsid of the Totavirus, the LRV virus, it protects the mice. And so something is going on, but we're a long way to know what it is. And also, is there any benefit for the leishmania? Because normally there's a playoff between the leishmania and between the virus and endosymbiont. They both benefit in one way or the other. Now, this is a very new player, we're in the game. Overwhelmingly, we're changing the environment in questions of evolutionary nanoseconds. This is unprecedented in any global event. It's almost equivalent to a KT event, which when that happened all those years ago. And the leishmania, I think they're quite happy. They're responding aggressively. We're having epidemics of visceral and cutaneous leishmaniasis. And if you look at it from the parasites' point of view, this is great because your numbers are increasing. You're increasing the areas where you are, and the size of your population. That's, I say, the leishmania population is increasing enormously. If we just take dogs, I mean, it's amazing. population of leishmania in dogs, if we counted it, I have no idea how to count it, but it's billions. I think with leishmania species come and go. And over the millions of years, I think there have been many, and we'll never know what they are. All we have are these that we have today. And I think man is really just a minute bubble in this evolutionary pathway, but he's very worried about it because it affects him so much. So my take home to you is that I think that the supercontinental hypothesis is the best one that explains very, very well the dispersion of the distribution of the leishmaniae genera and subgenera, and that the Pleistocene Ice Age was critical in molding the present ecogeography and diversity of the parasites we see today. Actually, I have slides that I took out and which show that this is the case with the Donovani complex and also with the major complex in the old world. This is Carl Sagan. I have great respect for him. He says, you have to know the past to understand the present. And I'm a great believer in this in all ways. Okay, well, thanks very much. Okay. What do I do now? Oh.
Hello?
Yeah.
Can you hear me, Jeff? Hello, Jeff?
Hello?
We have some questions.
Can you hear me?
Yes,
Oh,
of course.
dear.
I'm hearing you.
You're hearing me? Okay.
Okay, now
Yeah.
we're supposed to present to you some questions that have been made by the audience. Okay. The first one is from Hira Nagashi from FDA. He told, hi Jeff, fantastic talk. And again, he told you, how come there are no viruses in old world leishmaniasis, even though fungi in our prehistoric host were there? And then he told us, I just correct myself. In some major strains in Eastern Europe do have LRV. Any idea on how this happened?
Yeah, well, it's an ancient, it comes from the, well, first of all, I took one slide out, which shows that there are a lot of totoviruses have been found in major in Iran, a lot. And there's opening up a field there, the Iranians are. Now, it all goes back to the origin of these viruses are so old that they've come along with the ancestor, and they've just been passed on down the line all along the different phases of evolution, and the viruses have kept with us, kept with the leishmania. It's a very, but if you look at, I don't know, I have a slide, but if you look at the differences, it's quite amazing because the difference genetically is amazing. But let me go back to one of my slides. The difference between the leishmania, well, genetic differences, is sort of in the range of about, between the different genera of leishmania, is about, 90%, I think, 80%. They're that similar. The leash viruses are 50% different. The same virus in a leishmania. So they're evolving in a very different way. I think that's the message there. They're just evolving as all viruses do. They just divide, mutation. But the leishmania, they're not doing that. They're employing other methods of reproduction. And they have, I mentioned a number of them. Clonality is one, but also they have insect interchange, aneuploidy. So they're using a very different method, and so they're more conserved. The genetic variation in the leishmania is much more conserved than that of the viruses.
Okay, Jeff. Thank you. There is another question from Lucille Winter. And she's
Oh.
asking you, who could you say something about interspecific hybrids?
I could. Yeah. Okay. The work by David Sachs's group, I had a slide like that. I took that one out as well. They've shown that they reproduce, but the difference of future exchange between the F1 and the F2 is less. So you have, if you have intra-specific hybrids, they are more likely to exchange in the future, but at a less, a lower level than their mother strains. Now, in interspecies, they found it was very different, that the interspecies, they were sterile. They didn't actually cross. And when they tried to do the crossback, they didn't do the crossback. And that was between Major and Donovani. So I think if it's a close hybrid with a closely related species, it probably would survive. The ones between species that are way different, I think they will probably not survive. But you also have hybrid vigor, and that's been shown that one of the strains from Turkey, Peter Wolf, showed that it was more effective, and that was, I think, a hybrid between Infantum and Donovani, and that was more infectious to sandflies. So there was a hybrid vigor there in transmission.
Okay, Jeff. So there's some people that might make some questions. For instance, Philip Go, would you like to ask some questions to Professor Shaw? Jorge Alva, your opinions, guys, are always very
Georgie,
important.
hello. And what's he asked?
Okay, Jeff.
Yeah.
Jeff, do you really think that Leishmania chagasi and Leishmania infantum are the same species? No doubt about this.
Yes. Yes, I do. I do. Yes. I just think that they're the question. They're very close, but they're different. I mean, they've been shown that there are various. They've shown that they lack some genes, so they're different. And I think that there's evidence, some of your evidence and other clinicians, that they're more difficult to handle clinically than the classical infantum of the Mediterranean region. So they're infantums. I don't think I'd argue against that, but I think they're different. So I think maintaining the name Chagas is useful as a subspecies, maybe, or I think it draws attention to them being different, so it makes people look a bit more at them, and they're perhaps otherwise, oh, everything's infantum, so we don't look at it more. But if you give it a name, people get a bit cross sometimes, and they sort of say, oh, well, is it really? And they tend to delve into it more. And I think that's a good thing. But it's infantum, yes, it's infantum. But not exactly the same as the infantum of Iran, of North Africa, of France, of Spain, of Italy. It's different. Now, it's been imported many times, so that's another question. You could talk hours about this. Yeah.
Jeff, there is one question here from Yamuna Sirivardhana. She's asking you about, or she or he, I don't know exactly, what about Leishmania archibody?
Oh, well, what about them? I mean, yeah. Well, as far as in man, I think that at the moment, our big problem is that we base practically everything that we have and know on isolates from man. And we need to really know a lot more about isolates immediately from reservoirs. And I don't mean dogs, I mean reservoirs. because a dog, I think, is a bit like man. He's a bit of an accident. And they all, at least in dogs and man, they produce antibodies. In rodents, rodent models, they produce antibodies. The participation of antibodies in the successful immune response, I'm not an immunologist, and I wouldn't like to go into that area, but I think diagnostically they can be useful. I think this has been shown in Braziliensis in particular. People have from in Argentina, some very recent work have once again emphasized that Braziliensis produces antibodies, and it's a diagnostic tool to that level, really. Yeah, I think antibodies are a part of the immune response, but I wouldn't know how they participate or don't participate. You have to ask an immunologist that.
Okay, there's some more questions here, Jeff. First, to tell everybody that this presentation is going to be available in the website by YouTube, and you can see it anytime. It's quite interesting. So one of the best I saw from Jeff, although it's short, but it's pretty available. Later on, the organizer is going to send you more messages explaining exactly how to access to this presentation. Okay. And Nancy Saravia is asking Jeff, how important are humans as reservoirs in different scenarios for Leishmania infantum and dermatotropic species in the New World?
Yeah, good question. At the moment, there's not much evidence supporting them as being important reservoirs. I don't think they are. I think they're very much accidental. Maybe in certain domestic situations, man could be a source for Longipalpus, and I think you've shown that, haven't you, in Teresina. But I think his importance as a reservoir is not very great, very different from Tropica, where you have his importance there. Mediterranean area, maybe he's more important there than here. A lot of it depends on the relationship of the sandfly, the vector, with the reservoir. And I think in the old world, it is predominantly the sylvatic reservoirs for the cutaneous forms of the disease, and the dog, I think, for the visceral form. I mean, the number of dogs that are infected now, I don't know. It must be enormous. I have no idea.
Okay, so we have many questions, Jeff. Here from Dennis Sereno, asking evidence of hybridization events between Leishmania species infecting human are provided. Thus, we get information on other Leishmania species currently not having a medical interest. Thank you for the great speech.
Yeah, well, it is interesting, actually, that these hybrids have been isolated from patients. This is interesting. So they're going around in reservoirs. So they've survived. From the clinical point of view, I think it's very interesting. This is some work done in Canada by Greg and others, and they've just shown, it was published very recently. Some of you might know that they have cutaneous leishmaniasis in Sri Lanka, apparently caused by Donovani. Originally, they found that there were some differences in that Donovani. It wasn't exactly the same as the Indian Donovani. And Greg has just shown that actually the strains are hybrids between, some are hybrids between Tropica, and some are hybrids between major. how they got there. I mean, how they got there from Africa. One presumes that that hybridization between Donovani, Tropica, and Major must have occurred in Africa. But the result was that the disease is no longer cutaneous, no longer visceral. It's cutaneous, and that's in Sri Lanka. So it's a beautiful model, really. And this is very recent publication. I think Greg published it this year, and he used very sophisticated analysis to show this. And I was surprised at the results. But he shows that they are hybrids between Donovani, some Donovani and Tropica, and some Donovani and Major.
Okay, Jeff, by the way, HIV-infected patients, they have a more diverse leishmanial population than immune-competent patients.
Yeah.
Do you think that they can pose, say, a role in the dissemination, dispersion, or evolution of leishmania?
Yeah, I think so, because they're very unusual hosts in terms of, yeah, it's, in fact, some very, I think the people in Montpellier have isolated some very odd strains of infantum from HIV patients. I'm not too familiar with the work, but I do remember that they said that these strains from HIV patients were different. The potential, of course, is that they could become infected with multiple parasites, not just one, a number, and then a sandfly could pick up these multiple parasites, and you could get genetic exchange between different lines because the patient hasn't managed to respond and he has maybe been infected a number of times. And so I think that they potentially could be very important in terms of creating new lines in infantum. Yes, I think they could do, especially with vectors, when vectors come around. There's also the question of is there any sex exchange of amastigotes? That's a very difficult question. Very difficult. I wouldn't like to go into that, but some people think that there is. So if there's, and that's another area.
Okay, we have more questions, Jeff. We still have time for some, although not very much time. Jairo Mendonça Rodan, thanks you for your excellent presentation. He asks about the subgenus Sauroleishmania. There is a theory on the origin being from mammal leishmania. Could this mean that they could, in certain cases, in epidemiological context, infect mammals, such as the case of Leishmania alderi? And also, in your opinion, could reptiles be reservoirs of pathogenic leishmania? Interesting question.
I mean, yeah, it's an interesting question. And of course, the Adleri question, they've shown actually that they've isolated Adleri from man. So, but again, it's all comes down to the fly and the parasite. And the fact that the Sauroleishmania came from a mammalian line, it's not going too far back to go into a man. Now, I don't know that in China, they found Donovani infantum in snakes. So, you know, now, if that particular sandfly that bites snakes bit a man, it could infect a man, I suppose. It's a very fluid situation. And it all comes down to the ecosystem. That is what is controlling everything. It's because they're like the puppet masters. The ecosystem is the puppet master of the reservoirs and of the sandfly. That's what it is. That's why I place so much emphasis on the ecosystem, because it's what makes the difference, really. That's what it does. And we create ecosystems. I mean, man does. We've done a hell of a lot of damage to natural ecosystems. We made a lot of our own.
Yeah. Jeff, Hira comes back and asks you about Donovani causing cutaneous and visceral leishmaniasis. He asks you about the possibility that this is due instead of leishmania variation, but to the sandfly differences. What do you think about this?
Oh, I know this came up in Central America, in Honduras. Yeah, I don't think there's any evidence to support that. Let's put it that way. There's all the business of the saliva and all of the components and the different flies. But at the moment, the evidence suggests that it's the parasite that is the difference in the parasite. And an example of that is in Cyprus. In Cyprus, you've got both. You've got Donovani causing cutaneous leishmaniasis and Donovani causing visceral leishmaniasis. So I wouldn't put that. I just say that there's not much evidence to support that. It could be, but I'm not very convinced that there's evidence to support that.
Okay, let's go ahead. We still have time. From Andre Cutolo. He asks you about transmission of leishmania by sandflies was a kind of dogma. Midges are transmitting leishmania in Australia. Why midges don't transmit leishmania elsewhere? Can this be occurring out of Oceania?
Could you repeat that? That's a complicated question.
He asks you about midges transmitting leishmania in Australia. And then he asks if it's possible that other midges are transmitting leishmania in the other parts of the world.
Oh, yes. Yeah, I mean, I'll emphasize one point, that we have this group, the Moundinias, associated with cutaneous leishmaniasis in Nigeria, in cows and horses in Africa, Martinique, man. And we have no idea. The vector probably is a midge, a Ceratopongida. That's probably the vector, not a sandfly. And we don't have any idea what the reservoirs are. We have this group that, the Moundinias are a bit complicated because you have the group which includes the the parasite from Australia, and Henriette, and those, well, they're one line. And the other line, we don't have any idea. I think there are a lot of cases. And then you have in Thailand, the Moundinias causing visceral leishmaniasis there. Again, in Thailand, they don't have no idea what the reservoir is. So I think there's a whole lot of cycles going on that we have no idea about. And then suddenly, somebody finds them by luck or just by chance, somebody becomes infected. I think the situation in Thailand is fascinating. Why you've got this very ancient line causing visceral leishmaniasis, maybe the Thailand forests, I don't know. That's a nice thought. Maybe that was a, yeah.
Yeah, Felipe Danta Stories, by the way, is asking about if there are hidden species, hidden species of leishmania, like in the Amazon, for instance, that could show up again in fact humans. What's your opinion about this?
Oh yeah, there are. Yeah, there are. I mean, we've got some. I've got some data now that I'm trying to sort out, but I think there are other ones running around, yes, and definitely the interesting thing is that when things got bad in the ice ages, the animals and that retreated to sort of nicer places. The Amazon was one, Central America was another one, and the Guiana Shield was another one. And I think that there are a lot of things that the parasites that we discovered them in looking at animals, not at man, and then they were found in man. And people don't normally look at animals because it's difficult. So I think you get a bit of a biased view of the leishmania from man, because we think that our view of leishmaniasis is man, but it's not, it's animals. And so I think the bias that the way we look at leishmaniasis tends to be the parasites within man. And that's why I think it's important to say the word leishmanini, because there are a lot more who are not actually within the genus Leishmania that actually cause a disease that has been called leishmaniasis. So I might call it leishmaniasis, if you like, not leishmaniasis. But I don't think that'd catch on somehow. Maybe, I don't know.
Okay, Jeff, I'm going to
Yeah.
take advantage of my position here and ask you the question that the audience asked you, but there is one that I must ask you. You created of best media for communication in leishmaniasis, which is the leishmanial network by email. Would you tell us if it is still alive, how to get in there, and what are the most interesting subjects of discussion you got over there? Just a few words on leishmanial. To me, it's
well, unfortunately, it's sort of like the dinosaurs. I don't know if it's going to survive the periods now. It's not used much. I think some of the nicest ones that we have had are questions from clinicians about treatments, you know, looking people who haven't dealt with this particular parasite, asking for help, you know, what can we do? I think that's probably one of the most important things. Doesn't happen very often, occasionally it does. But I think on the whole, people in leishmaniasis are very much very reclusive. They don't talk much to each other, and they don't say much about what's going on. It would be I'd like them to more, but initially it was used quite a lot. But I suppose other means have come about, but it's still alive. And if anybody wants to ask a question, they can send one, and it'll be circulated to about 4,000 people who are still there. About 4,000 people there.
4,000 is a lot. It's a lot.
Yeah,
Jeff,
quite a lot.
Jeff,
yeah.
Thank you a lot. I'm going to give the word to the organizer, Ivan Valles, and must tell people why this guy is really the great scientist that he is, with a wide view of science. He's a pure scientist, in the sense that I desire to have been one day, but I'm just a physician. So good luck for you, Jeff.
Carlos, you're good. You are a very good scientist and doctor, both.
Okay, thank you for the word. Ivan, the word is on. The word's on. Bye-bye, Jeff. Thank you a lot.
Okay. Bye-bye. Okay.
It's up to you, Ivan. Yami.
Gracias, doctor Costa. Gracias, doctor Jeff.
Now I have to remember you guys that in September, we're going to have the 6th webinar with Dr. Elisa Cupolillo. She's really great. She's going to take the point of Jeff to explore the genetics of leishmania with the species diversity or diversity of names, interesting name, pattern hidden behind leishmania taxonomy. So as I told you, the speeches are available to everyone via the internet. And Jeff, again, thank you. And Elisa, we're waiting for you. Bye-bye.